Welcome to the molecular markers module. So I'm going to start by using an example that is based on my own research. So as I believe I mentioned before, a lot of research in my lab focuses on this intertidal snail shown here, Chlorosoma funnebralis. And this species is distributed all along the western coast of North America. So that's what's shown here. They're found from Alaska in the north all the way down to Baja, California in the south. So that spans about 1,800 kilometers. And so what I want you to think about is, given how big this range is whether you think that snails collected from different geographic sites. So when we say geographic sites, we mean northern versus southern California, or like I said, Alaska versus Baja. So that's what I mean by different geographic sites. Do you think snails collected from different geographic sites are genetically different? And so this is, of course, based on that teaser I gave you at the end of the last module, where I used that picture of Gary the snail and said that we were going to have some conflicting results. So first I want you to think about what you think and whether you think snails collected from these different geographic sites are genetically different. Of course, in this picture, this is not a snail. That's my dog Enzo for scale. And then this is the snail here, in case that wasn't already clear. So I'll give you a second to think about what you think is the answer to this question. And so the answer really is whether or not, it depends on what study you look at. So like I said, there are some conflicting results. So what I'm showing here is taken from a paper that was published in 2010. And this paper says that the snail Tegula funnebralis, don't worry about this, they've changed the name a couple times, it's the same snail. The snail shows practically no structure from Oregon down to Southern California. So this part, no structure, that just means that all individuals... My dog Enzo is groaning in the background. I showed a picture of him, I thought he would be pleased. All individuals are genetically the same. So that's what that paper found, that based on their data, all the snails, whether you get them in Oregon or all the way down to Southern California, they're the same. However, a different paper that I published in 2016 says that we identified loci, so loci is another term for a genetic marker, that show evidence for regional differences between Northern and Southern California. And so based on this paper, we would say that Northern and Southern California are genetically different. And so if you're wondering, well, how can that be? How can both of these things be true at the same time, or who was right? What this example illustrates is that it depends on what type of molecular marker you use for your study that will affect the type of result that you get. So these two studies that I just mentioned were actually based on different molecular markers. So we're going to come back to this example a couple times throughout the module to illustrate how important that is in terms of marker choice, and also really to illustrate how the technology has advanced within the last decade or so within the field. So we'll come back to this example later, but just to set the stage. In this module, we have a couple different learning objectives, so these are things you will be able to do by the time you complete this module. We'll, first of all, compare and contrast different types of markers. We'll mostly accomplish this with the video quizzes. So, like before, I'm going to have a very short quiz, 1 or 2 questions after each video for you to take to apply what you just learned. And then the last two modules here, last two learning objectives focus on selecting a type of marker to use for a given study and then explaining or articulating why that marker is the best choice. And we're mostly going to accomplish these last two objectives through the main assignment for this module, which you will complete after watching each of those videos and taking the little mini quizzes embedded within there. And then at the end of the module, we'll put all of that information together and do an assignment that will give you practice choosing what molecular markers to use. So I'll basically give you different scenarios and ask you what type of marker would you use to address this question and why. So if we think of these doors here I'm showing can represent different options. So this option might be SNPs as a molecular marker. This might be microsatellite. We'll talk about all of these throughout the module, or this might be something like a high throughput sequencing marker. So we'll do an assignment at the end of the module to help you practice this. And why I'm asking you to do this is going to help you succeed in the class. So again, remember one of our main assignments at the end of the semester is going to be writing one of these GRFP style proposals. And in order for you to write that proposal, you need to decide on your own what type of marker to use. So we're going to start getting some practice with that to start building towards that final product for our class.